Imagine waking up one morning completely paralyzed. A spinal cord injury devastates not just the body, but the dreams and independence of millions worldwide. For decades, such injuries were considered permanent, irreversible, death sentences, basically. The spinal cord was thought to be incapable of healing itself, locked in a prison of scar tissue and inflammation that left people condemned to paralysis. But something extraordinary is happening in labs and hospitals around the world. Scientists are learning to speak the language of the nervous system itself, and they are discovering that the brain and spinal cord have far more regenerative potential than we ever imagined. We are standing at the precipice of a revolution that could transform what it means to recover from neurological injury, and breakthrough discoveries emerging in 2025 are nothing short of remarkable. So, today on Knowing is Winning, I will talk about the regeneration revolution and how science is rewiring the human nervous system. Well, rewriting and rewiring, actually. Anyway, at the heart of this revolution lies an elegant biological truth that researchers are finally harnessing. The nervous system can be rebuilt. In April 2025, Keio University Hospital in Japan completed a groundbreaking clinical study that had shaken the foundations of neuroscience. The research involved using iPS cells, which stands for induced pluripotent stem cells, essentially reprogrammed adult cells that can become almost any cell type in the body. Scientists took these iPS cells, carefully differentiated them into neural stem and progenitor cells, and transplanted them directly into patients with severe spinal cord injuries. The results were stunning. In one case, a patient who was completely paralyzed, classified as grade A on the American Spinal Injury Association scale, improved to grade C, meaning they regained significant motor function. Another patient improved from grade A to grade D. These weren't marginal improvements. These were patients recovering meaningful movement in a condition that is universally understood as permanent. The genius of this approach lies in the understanding what neural stem cells actually are. These are immature cells from the nervous system capable of doing two things. They can renew themselves indefinitely, and they can differentiate into any major cell type of the nervous system: neurons, astrocytes, and oligodendrocytes. When transplanted into an injured spinal cord, they don't just become replacement cells. They transform the entire microenvironment around the injury. The tissue around spinal cord damage is hostile to healing. Inflammation rages unchecked. Scar tissue accumulates like a wall, preventing regeneration. Reactive oxygen species attack healthy tissue. But neural stem cells act like peacemakers. They release anti-inflammatory molecules. They reduce dangerous compounds that damage neurons. They promote remyelination, the process of rebuilding the protective sheaths around the fibers that allow electrical signals to travel at full speed. The Keio study involved only 4 patients, but the statistical significance is extraordinary. In comparable patients tracked through the Japan Spinal Injury Center database, the median improvement in motor function at 52 weeks was between 4 and 7 points. The iPS cell transplant patients improved by a median of 13 points, nearly double. More remarkably, only 10 to 12% of untreated spinal cord injury patients naturally improve to grade C or higher. In this small group receiving the transplant, 50% achieved that level of recovery. These numbers suggest something that many neuroscientists thought was impossible. The severely injured adult nervous system can be convinced to rebuild itself. But chaos breakthrough is just one piece of a much larger transformation happening in neuroscience. Scientists worldwide are discovering multiple pathways to neurodegeneration, each one opening new possibilities. At UC Davis, researchers identified two compounds called K102 and K110 that could reverse nerve damage from multiple sclerosis. Multiple sclerosis destroys myelin, the insulating layer that allows neurons to communicate rapidly and efficiently. Without it, signals move sluggishly or stop entirely, leading to progressive paralysis, vision loss, and cognitive decline. For decades, MS treatments focused on suppressing the immune system to reduce inflammation. But these new compounds do something revolutionary. They actually rebuild the damaged myelin. They promote, if you would, remyelination. They reactivate the body's own repair mechanisms. Licensed by Cadenza Bio, these compounds represent a leap from lab curiosity to potential clinical therapy. The implications cascade outward. At the University College London, researchers identified a crucial molecule called CCL3 that acts as a directional signal guiding Schwann cells across nerve injury sites. Schwann cells are the builders of peripheral nerve regeneration. They wrap around axons and speed up signal transmission, basically. Anyway, the scientists discovered that CCL3 acts like a chemical compass, secreted by immune cells at the injury site, telling Schwann cells exactly where to go and how to rebuild the nerve bridge. When researchers blocked CCL3, regeneration stalled. When they enhanced it, repair accelerated. This single discovery opens pathways to enhance recovery from traumatic nerve injuries, from accidents that sever limbs to injuries from burns or surgery. At UC San Diego, a different approach entirely yielded surprising results. Researchers used advanced bioinformatics and gene sequencing to comb through databases of existing drugs looking for compounds that could enhance neural regeneration. They discovered thyrophan, a medication previously tested in humans for entirely different conditions. Apparently, it could dramatically stimulate nerve self-growth. When they tested it in rats with spinal cord injuries, the results were striking. Rats receiving thyrophan alone showed 50% improvement in hand function. When combined with neural stem cell transplants, they achieved an additional 50% improvement, effectively doubling the recovery benefit. Because thyrophan was already safely tested in humans, it could enter clinical trials for spinal cord injury far more rapidly than drugs still in early development stages. Then, at NerveGen Pharma, clinical trials of a drug called NVG-291 showed that patients with spinal cord injuries sustained for 1 to 10 years, well, basically injuries considered chronic and hopeless, could still recover meaningful motor connectivity and function. This shattered the fundamental assumption in neurology, that there was a narrow therapeutic window, that after a certain point the nervous system couldn't be repaired. The chronic SCI patients receiving NVG-291 showed improved motor connectivity compared to placebo, demonstrating that even years after injury, the nervous system retains the capacity to reorganize and strengthen functional pathways. The FDA has granted NVG-291 Fast Track designation because the data is so compelling. The deeper we peer into neural regeneration, the more we discover that the brain and spinal cord have been hardwired for repair all along. We just didn't know how to activate these mechanisms. Neuroplasticity, the brain's ability to reorganize itself by forming new neural connections, was once believed to be confined to childhood development. Scientists thought that after reaching adulthood, the architecture of the brain was fixed and immutable. This belief dominated neuroscience for over a century. But it was wrong. Research throughout the 2020s has definitely shown that neuroplasticity continues throughout the entire human lifespan. The brain remains plastic, moldable, capable of rewiring itself in response to experience, injury, learning, and therapy. Physical activity is emerging as one of the most powerful activators of neuroplasticity. Exercise doesn't just build muscles, it transforms the brain. When you exercise, particularly through high-intensity interval training, your body floods neural tissue with brain-derived neuroprotective factor, or BDNF. BDNF is like fertilizer for neurons. It promotes synaptic plasticity, the strengthening and weakening of connections between neurons that underlies learning and memory. BDNF drives neurogenesis, the birth of entirely new neurons, particularly in hippocampus, the brain's memory center. Aerobic exercise, resistance training, and high-intensity interval training all upregulate BDNF expression, effectively rewiring the brain for enhanced cognition, memory, and neuroprotection against disease like Parkinson's and Alzheimer's. In one remarkable study, researchers showed that aerobic exercise increased hippocampal volume in older adults and improved their cognitive function. They weren't just maintaining brain health, they were growing new brain tissue. In other studies, HIIT enhanced the effectiveness of non-invasive brain stimulation, suggesting that when exercise primes the brain for plasticity, other interventions become more potent. This has profound implications for rehabilitation. A person recovering from stroke can exercise intensely to trigger BDNF release, enhancing their brain's responsiveness to physical therapy. A patient with Parkinson's disease can combine exercise with targeted neuromodulation to accelerate neural adaptation and recovery. But exercise is just one lever. Pharmaceutical interventions are opening other pathways. Phenelzyme, an antidepressant medication, is showing unexpected promise for spinal cord injuries. When the spinal cord is damaged, secondary damage cascades through the tissue. Oxidative stress ravages cell membranes. A particularly destructive compound called acrolein accumulates to toxic levels, damaging structural proteins and enzyme systems. It's like a chemical fire consuming healthy tissue around the injury. Phenelzyme acts as an acrolein scavenger, binding to it basically and neutralizing its reactivity. When researchers administered phenelzyme to spinal cord injured patients, pain decreased and movement improved. It demonstrates the principle that understanding the molecular machinery of injury allows us to develop precision interventions. One of the most exciting discoveries is emerging from Hong Kong University of Medicine. Researchers identified peripheral neural stem cells, PNSCS, hiding in mouse limb and lung tissues. For decades, scientists believed that neural stem cells existed only in the brain and spinal cord, tucked deep inside tissue where they were difficult to access. But this discovery suggests these regenerative cells may exist throughout the body. If similar cells exist in human tissues and can be cultivated, they could revolutionize treatment approaches. Imagine being able to harvest regenerative neural cells from your own limbs and expand them in culture for transplantation back into spinal cord injuries or neurodegenerative diseases. It sidesteps immune rejection, eliminates ethical concerns about embryonic cells, and provides a ready source of regenerative potential. Building on this principle of harnessing the body's own regenerative capacity, scientists at universities worldwide are developing induced neural stem cells, or INSCS, directly from patients' blood cells or skin cells. This shortcut bypasses the lengthy process of creating iPS cells and then differentiating them into neural cells. Instead, researchers directly reprogram patient cells into neural stem cells using viral vectors carrying modified genes. The process is 4 times faster than traditional approaches and dramatically reduces costs and time and culture. Once created, the INSCSs reduce neuroinflammation and restore motor control in animal models of multiple sclerosis and spinal cord injury. They recognize and absorb inflammatory molecules like toxic succinate from cerebral spinal fluid, effectively detoxifying the injury site. They activate anti-inflammatory genes that promote remyelination and allow surviving neurons to form new connections. The clinical implications are becoming clear. The future of neurological medicine will not be a single cure-all, but rather a symphony of complementary approaches. After a spinal cord injury, a patient might receive acute pharmaceutical intervention to reduce secondary damage, perhaps a drug like phenelzyne or thyrofan to protect remaining tissue. Within the therapeutic window of days to weeks, they might receive neural stem cells or progenitor cells transplantation to rebuild damaged tissue and restore connectivity. Simultaneously, they would engage in intensive physical therapy coupled with high-intensity exercise to maximize neuroplasticity and activate brain-derived neurotrophic factor. At later stages, they might receive pharmacological agents like NVG-291 that promote regeneration and network reorganization, even years after injury. Brain-computer interfaces might restore motor control by bypassing damaged spinal pathways entirely, creating new technological conduits for the brain's intentions to drive movement. This multimodal approach reflects a fundamental shift in thinking. We are moving from a paradigm where injury means permanent disability toward one where injury triggers a cascade of biological and technological interventions that harness the nervous system's own regenerative potential. The spinal cord no longer appears as an immobile, unchangeable structure, but as a dynamic tissue capable of astounding repair and rewiring when given the right signals and support. The revolution extends beyond spinal cord injury. Multiple sclerosis patients face deteriorating myelin, but compounds like K102 and K110 offer the possibility of rebuilding what disease has destroyed. Stroke victims suffered massive cell death and disrupted neural networks, but rehabilitation enhanced with exercise, pharmaceutical support, and potentially cell transplantation could allow the surviving brain tissue to rewire and recover function that seemed lost. Parkinson's disease and Alzheimer's disease, long considered neurodegenerative death spirals, might be slowed or even reversed through combinatorial approaches that enhance neuroplasticity and neurogenesis while simultaneously protecting remaining neurons. Beyond injury and disease, these discoveries about neuroregeneration illuminate the brain's capacity for enhancement. If neuroplasticity is lifelong, if BDNF can be upregulated through exercise and other means, if neural stem cells can be generated from our own tissue, then the possibilities for healthy aging and cognitive enhancement expand dramatically. Older adults could engage in structured exercise and neural challenges to maintain cognitive sharpness well into advanced age. Professionals seeking to master complex skills could potentially enhance their neuroplasticity through targeted interventions. The boundary between treatment of disease and optimization of human capabilities blurs. What we are witnessing in 2025 is the end of an era of neurological determinism. The belief that nervous system is fixed and immutable after development. We are entering an era of neural possibility. For millions suffering from spinal cord injuries, multiple sclerosis, strokes, and neurodegenerative diseases, these breakthroughs represent genuine hope. For all of us interested in the remarkable plasticity and regenerative capacity of the human nervous system, this is a time of extraordinary scientific progress. The brain and spinal cord are not dead tissue. They are living, dynamic, regenerative systems waiting for us to understand how to unlock their potential. And the potential appears far greater than we ever imagined. Knowing is winning, and now we are winning the right to know the true regenerative power of the human nervous system. Thank you very much for watching.